Hello everyone, welcome back. Today I'm here to discuss about the introduction of plant physiology. So what is plant physiology and what are the various aims and objectives of plant physiology and what are the various applications of plant physiology in the field of agriculture and horticulture. So these are the topics of discussion that I will try to cover in this presentation. Let's start. So firstly, what is this plant physiology? Physiology word is derived from a Greek word physis, which means nature, and logos means discourse or study. So these two Greek words, that is physis means nature, and logos means discourse or study. So this is basically the study of various functions and processes which are occurring in the plants. तो plants के अंदर जो भी life processes और functions होते हैं उनकी study को हम physiology बोलते हैं. So it is a science that studies plant functions. What is going on in plants that accounts for their being alive, जिसकी वजह से हम उन्हें बोलते हैं कि plants alive है, living है. So this definition was given by Salisbury and Ross in 1992. इनकी एक बुक भी आती है plant physiology की. And second definition is by Theis and Ziegler. So the study of plant function basically that encompassing the dynamic processes of growth, metabolism and reproduction in living plants. So जो भी ये processes होते हैं और functions होते हैं plants में like growth, metabolism and reproduction, इनकी study को हम कहते हैं plant physiology. Then who is the father of plant physiology? So this is the person named as Stephen Hales, who is considered as father of plant physiology, and he was the first person to demonstrate that or to explain that the minerals, salts and water is connected by xylem in plants and the organic matter is connected by phloem in plants. So he was the first person to explain this division of liver in plants. And this is the portrait of the person, that is Sir J.C. Bose, who is considered as father of Indian plant physiology. And this term physiology was coined by Jean Fernel in 1525. So he was basically a French physicist who coined this term plant physiology. That is Jean Fernel. Next, there are some other definitions of plant physiology. Basically, this is the sub branch, or we can say branch of botany that deals with the life processes of plants. Then there's one journal who explained this in another way that I will discuss. So these life processes include photosynthesis, transpiration. So photosynthesis is a process by which plants capture the solar energy and convert it into chemical form of energy by using substrates like carbon dioxide and water to produce sugar that is utilized by plants, and in a result of this reaction produces oxygen that is utilized by other aerobic organisms. The next, transpiration, that is loss of water from the aerial part of the plant in the form of water vapors. And water relation, how the plant conducts water from soil to the different plant parts. And respiration, that is the breakdown of this glucose and release of energy that is utilized by the plants. Then biosynthesis and functions of phytohormones, how plant hormones are synthesized and what are the various functions of these plant hormones. Then next, study of plant movements, photoperiodism, that how this day duration affects the plant, and flowering in plants. Then seed germination, how seed germinates and produces the plant. Then there's a definition which is given by a journal, which is a very famous journal, Nature. So according to this journal, this plant physiology is a subdiscipline concerned with subdiscipline of botany basically. Botany का एक subdiscipline है that is concerned with the physical, chemical and biological functioning of the plant, जिसमें physical, chemical और biological functioning को हम study करते हैं plants की. And there is another definition. So this is a study of vital processes and functional activities occurring in the plants. This is again the definition of plant physiology. So जो भी vital processes, important processes होते हैं plants में, and functional activities जो plants में होती हैं उसकी study को हम कहते हैं plant physiology. Next comes aims and objectives of plant physiology. So what are the basic aims and objectives of plant physiology? Basically, the knowledge of these processes can help us to improve our crops, to get more yield, to make plant disease resistant, and various plant products that we can get to increase our economy and to fulfill our needs. So that's why we need to study plant physiology. Firstly, we can observe molecular, physiological, and biochemical mechanisms of vital processes. So this is the main objective of this branch, that is plant physiology. By observing these phenomena, we can alter the plant mechanisms and can get maximum benefit. Next, we can study genetic, physiological, and enzymatic mechanisms which occurs during growth. जब प्लांट ग्रो करता है उसमें कौन कौन से जेनेटिक चेंजेस आते हैं, फिजियोलॉजिकल चेंजेस आते हैं और एंजाइमेटिक एक्शंस होते हैं, उनको हम स्टडी कर सकते हैं. Then next we can elucidate or explain the mechanisms in response to external environment, how plants responded to external environmental conditions. जो environmental stimulus है उसमें plant कैसे खुद को respond करता है और कैसे adapt करता है. The next study of mechanism of nitrogen fixation, how plants fixes atmospheric nitrogen and utilize this nitrogen. तो ये भी हम study करते हैं इसमें. Next, we can study mechanism of nutrition, growth, movement, and reproduction. How plants get its nutrition, how it grows, and how it shows some movements and how it reproduces. So ये सारी studies हम करते हैं plant physiology में. The next mechanism that control gene activity during ontogenesis. So how genes expresses themselves during ontogenesis. Ontogenesis means the study of various developmental stages, embryonic developmental stages. जो प्लांट की एम्ब्रियो की डेवलपमेंटल स्टेजेस होती हैं उनकी स्टडी हम कहते हैं ontogenesis. The next we can study the mechanism of biosynthesis of some secondary metabolites like alkaloids, rubber, phenolic compounds. These secondary metabolites are not directly utilized in the plant metabolism. Indirectly plant metabolism में use नहीं होते हैं, like with that of glucose or we can say carbohydrate, proteins or fats. They are produced in addition to these metabolites and they play a very important role in the plant defense, protection and healing the plant wounds. Next, plant responses. We can study plant responses and resistance to environmental stresses. So how plant respond to environmental stresses, how it gets resistant to the environmental stresses. So these mechanisms we can study under this branch of botany, that is plant physiology. And we can also discover the function of plant growth regulators, means जो plant hormones हैं उनके क्या functions हैं और उनको हम कैसे अपने agriculture और horticulture में use कर सकते हैं. Then we can use these PGRs in agriculture and horticulture. So these are main aims and objectives of plant physiology. Next comes the application of plant physiology in agriculture and horticulture. So firstly, what is agriculture? Agriculture is a science and art of cultivating soil, growing crops and rearing livestock. मीन्स जिसमें हम सॉइल कल्टीवेट करते हैं, क्रॉप्स को ग्रो करते हैं और जो पालतू जानवर है, डोमेस्टिक एनिमल्स हैं, उनको रियर करते हैं, उनका केयर करते हैं. सो दैट ब्रांच इज नॉन एज एग्रीकल्चर. इन हॉर्टिकल्चर वी mainly deal with this in the science and art of growing vegetables, flowers and fruits. So firstly, the main purpose of this plant physiology is to enhance crop yield, crop production, crop जो फसल की पैदावार है उसको increase करना है. So this is the main objective. So firstly, as we already know that plants are dependent upon some basic elements which plays major important role or major role in their main normal growth, that is carbon and nitrogen. So by using these two elements or by using the mechanism how plant utilizes these two elements, we can get maximum yield. In addition to this, this nitrogen is very important component in plant growth and it acts as a limiting factor. मीन्स अगर सारी चीज़ें अवेलेबल है सॉइल में बट नाइट्रोजन नहीं है, सो प्लांट की ग्रोथ नॉर्मल नहीं होगी. सो दैट्स व्हाई दिस इज लिमिटिंग फैक्टर फॉर नॉर्मल ग्रोथ एंड फंक्शनिंग ऑफ द प्लांट. सो कार्बन एंड नाइट्रोजन आर द मेन कॉन्ट्रीब्यूटर्स टू क्रॉप यील्ड. क्रॉप की यील्ड कितनी होगी, ये दो फैक्टर्स हैं जो डिटरमाइन करते हैं क्रॉप यील्ड को. देन नाइट्रोजन इज ऑलवेज अ लिमिटिंग फैक्टर, मीन्स ये हम देख पाते हैं नाइट्रोजन की डेफिशिएंसी है तो प्लांट नॉर्मली ग्रो नहीं कर पाता है. So we have some important microbes जो कि नाइट्रोजन को फिक्स करते हैं और प्लांट को प्रोवाइड करते हैं. प्लांट्स में ये कैपेबिलिटी नहीं होती है. सो वी हैव सम नाइट्रोजन फिक्सिंग बैक्टीरिया विच में बी सिम्बायोटिक मीन्स एसोसिएशन में फिक्स करते हैं या फिर एसिम्बायोटिक और वी कैन से फ्री लिविंग जो freely नाइट्रोजन को फिक्स करते हैं, like we have some aerobic bacteria like Azobacter, Azospirillum, Klebsiella. Then this next bacteria is facultative bacteria. It means it is not fully aerobic. It can grow in under aerobic conditions also. So this is Klebsiella and Bacillus. The next we have anaerobic bacteria, which are photosynthetic, means they can synthesize their food. That is Formatium, Chlorobium, Clostridium. Then we have some cyanobacteria, like we have Anabaena and Nostoc. So these microbes have capability to fix atmospheric nitrogen into that form that can be utilized by plant. तो ये atmospheric nitrogen को fix करते हैं और ऐसी form में convert करते हैं, like we have this that is nitrate ion. इस form को plant absorb करते हैं. So use of nitrogen fixing bacteria in agriculture land. So we can use these nitrogen fixing bacteria. We can basically get the culture of these bacteria, and we can mix our seeds with these bacterial culture and can sow in the field. तो हम क्या करते हैं, ये जो seeds होते हैं उनको हम mix करते हैं बैक्टीरियल इनोकुलम के साथ, जो नाइट्रोजन फिक्सिंग बैक्टीरिया है, एंड आफ्टर दैट वी सो दैट इन फील्ड, और वहाँ पे ये जो माइक्रोब्स हैं ये नाइट्रोजन को फिक्स करते हैं जिससे प्लांट की ग्रोथ. Next, we have some symbiotic nitrogen fixing bacteria like we have Rhizobium. These are mainly associated with legumes. जो दालें होती हैं उनके साथ associated होते हैं legumes के साथ, pulses के साथ. Then we have Bradyrhizobium. This Bradyrhizobium japonicum is a microbe that is associated with one crop, that is Glycine max. Glycine max, and this Glycine max is the scientific name of soybean. जो सोयाबीन है उसका एसोसिएट होता है. वी हैव दिस Azorhizobium. The next comes how we can identify these bacteria or microbes in the plants. So basically what they do in plant roots, they form these bead like structure which are known as nodules. इन्हें nodules बोलते हैं. इन nodules में ये living ये जो microbes हैं inhabit करते हैं, ये means यहाँ पे रहते हैं, and they produces the outgrowth which are known as nodules, और ये यहीं पे fix करते हैं atmospheric nitrogen और plant को nitrogen provide करते हैं. If we cut the section of this nodule, we observe a pinkish color substance in this section, और वो जो pink color का substance होता है उसे कहते हैं lag hemoglobin. Basically ये जो function है lag hemoglobin का, it provide oxygen free environment for the microbes, or the enzyme which is used in nitrogen fixation, that is nitrogenase. जो nitrogenase enzyme होता है उसको नॉर्मल फंक्शनिंग के लिए चाहिए होता है ऑक्सीजन फ्री एनवायरनमेंट. सो लैग हीमोग्लोबिन का काम है ऑक्सीजन फ्री एनवायरनमेंट प्रोवाइड करना इस एंजाइम के लिए. नेक्स्ट वी हैव सम asymbiotic bacteria like Azospirillum, Azobacter and Frankia. These are free living nitrogen fixing bacteria. So by altering the pathways जिसमें कार्बन और नाइट्रोजन इन्वॉल्व होते हैं, इन पाथवेज को अल्टर करके हम by using biotechnological tool we can enhance or we can improve the crop yield, जिससे हम फसल की पैदावार को improve कर सकते हैं. The next important application of plant physiology is by using certain PGRs we can induce rootings in stem cuttings so that we can multiply the plants in shorter durations. So basically we can use some stem cuttings and we can treat these stem cuttings with some suitable PGRs, like we have IBA, that is indole butyric acid, and this chemical helps in formation of roots or inducing roots in stem cuttings so that we can multiply the plants. Here we can see this is a hibiscus stem cutting, and by treating with this chemical substance or PGR they can produce roots and can produce multiple plants. So horticultural plants can be multiplied through rooting of the stem cuttings, and for that we can use these chemicals like indole butyric acid. So this chemical can induce the rooting in these stem cuttings. This is the rooting compound, that is IBA, rooting hormone. So we can use this in multiplication of these plants. Next important application of plant physiology is flowering induction. Means we can use certain chemical substances or PGRs to induce flowering in certain plants. We have some common hormones like GA and IAA, that is gibberellic acid and indole acetic acid. So these chemical substances or plant hormones can induce flowering in certain plants. So induced through application of gibberellic acid in some plants, and also be enhanced by using indole acetic acid or succinic acid dimethyl hydrazide. So these are some PGRs that can be used to induce the flowering in certain plants. इससे हम कुछ plants में flowers को induce कर सकते हैं, और they can be established in the plants so that we can get good yield of fruits. The next important application is flowering at will in floriculture. So this is very important. By the knowledge of plant physiology, especially the photoperiod and vernalization, दो important चीज़ें हैं. अगर हमें इसका proper knowledge है, so we can produce the flowers at our will. Means off seasons के flowers हम produce कर सकते हैं, and we can get higher price. So off season flowers gives higher returns to the farmers. और इसके लिए हमें knowledge होना चाहिए photoperiodism का और vernalization का. So photoperiod, as the name indicate, photo means light, period means duration. So एक दिन में average जो light है किसी plant की requirement to induce flowering is known as the photoperiod, and that minimum requirement is known as critical day length. So knowledge of photoperiodism and vernalization. So vernalization is a term that is used for chill treatment और लो टेम्परेचर ट्रीटमेंट टू इंड्यूस फ्लावरिंग प्लांट. सो कुछ प्लांट्स में चिल ट्रीटमेंट चाहिए होती है फ्लावरिंग इंड्यूस करने के लिए. सो दैट इज कॉल्ड वर्नलाइजेशन. सो दीज टू कंसेप्ट्स कैन बी यूज्ड so that we can produce flowers at our will. So floriculture crops can be induced to flower at will of farmer by controlling light and temperature. So ये जो चीज़ें हैं इनको कंट्रोल करके हम flowering induce कर सकते हैं. Like we have one example, that is this plant. This is very important ornamental plant, chrysanthemum, that can be induced to flower in summer by cutting short light exposure. So अगर हम इस plant में basically ये जो plant है ये autumn में flowers produce करता है. So we can make it this to flower in summer condition, but what we have to do, we have to reduce the day length for 15 days. 15 days के लिए हमें इसे short day duration में रखना पड़ेगा, and after that it can produce flowers. So this is the proper knowledge of photoperiodism, and we can induce flowering in this plant, जो कि basically autumn में flower करता है, but we can induce this plant to produce flowers in summer conditions, spring condition. So this is the photograph of this plant, that is chrysanthemum. Next, the knowledge of plant physiology can help in fruit setting and fruit enhancement. So we can use certain PGRs, like we have IBA, that is indole butyric acid, then 4-chlorophenoxyacetic acid, that is 4-CPA, abbreviated as 4-CPA. Then this is very important. Plant growth regulator, that is 2,4-dichlorophenoxyacetic acid, abbreviated as 2,4-D. Then gibberellic acid, chloramquat chloride, CCC, abbreviated as CCC. Then succinic acid dimethyl hydrazide, or triiodobenzoic acid, this TIBA. So these chemicals can be used to enhance the fruit set. जो फ्रूट सेटिंग है उसको ये एनहांस करते हैं. दिस टिबा इज़ वेरी इंपॉर्टेंट केमिकल कंपाउंड. इसे हम एंटीऑक्सिन भी कहते हैं. इसकी जो फंक्शंस होते हैं, ये एंटीऑक्सिन है, मींस ऑक्सिन की सिंथेसिस को ये रोकते हैं. सो दिस इज़ नोन एज एंटीऑक्सिन. मींस ये काम करता है एंटागोनिस्टिकली, स्टॉप करता है ऑक्सिन की फंक्शनिंग को. नेक्स्ट इम्पोर्टेंट एप्लीकेशन इज़ प्रिवेंशन ऑफ फ्रूट ड्रॉप. ये एक बहुत ही कॉमन प्रॉब्लम है दैट फार्मर्स फेसेस. सो इस प्रॉब्लम को भी हम सॉल्व कर सकते हैं बाय यूजिंग दिस knowledge of plant physiology and by applying certain PGRs. So fruit drop is a very common problem that every farmer faces. So major problem can be prevented by application of this chemical, that is IBA, naphthyl acetic acid, NAA, and 2,4-dichlorophenoxyacetic acid. So these chemicals can be used to prevent this premature fruit drop, and these chemicals are crop specific. ऐसा नहीं है कि universally सभी crops में use होते हैं. These PGRs are mainly crop specific. Then fruit thinning. Sometimes अगर फ्रूट ज़्यादा क्राउडेड होंगे, बहुत ज़्यादा इनका नंबर होगा, सो जो फ्रूट्स क्वालिटी है वो रिड्यूस हो जाती है और फ्रूट का साइज़ भी रिड्यूस होता है. सो टू गेट गुड क्वालिटी फ्रूट और हैविंग अ गुड साइज ऑफ दैट फ्रूट, वी हैव टू रिमूव सम फ्रूट्स फ्रॉम this cluster so that the fruit size becomes best of its quality. For that we can use certain chemicals, so useful to increase fruit size. Fruit thinning ज़रूरी है size को improve करने के लिए. So this is a chemical that is 4-CPA, that is 4-chlorophenoxyacetic acid. So this chemical can be used. इसे कार्बाइल के नाम से भी जानते हैं. दिस कैन बी यूज फॉर फ्रूट थिनिंग, मीन्स ये फ्रूट थिनिंग में हेल्प करता है, कुछ फ्रूट्स ड्रॉप हो जाएंगे और जो बचेंगे दे विल गेट ऑल द न्यूट्रिएंट्स एंड हैव गुड साइज और क्वालिटी. नेक्स्ट इंपॉर्टेंट फीचर और एप्लीकेशन इज डिज़ायरेबल फूड साइज एंड कलर. सो इसके लिए भी हम कुछ PGRs को यूज़ करते हैं जिससे हमें एक डेफिनेट साइज़ और कलर फ्रूट का मिलता है किसी फार्मर को, एंड ही कैन गेट मैक्सिमम मनी और we can say maximum benefit from this crop. So GA is used to increase fruit size. This is very important function of GA. Ethylene, that we already know this ethylene or ethyl, and its trade name is ethefone, जो कि fruit ripening और fruit coloration में help करता है. Then next is fruit ripening. So this is the chemical ethylene or 4-CPA then SADH. So these chemicals are used in fruit ripening. Basically the most common one is this gaseous hormone that is ethylene. ये mostly fruit ripening में help करता है. Then we have some hormones like IAA. ये fruit ripening को prevent करता है, means fruit को ripe नहीं होने देता है, so that's why it can increase the shelf life of fruit, means लंबे समय तक हम fruit को store करके रख सकते हैं. So this is एंटागनिस्टिक टू इथाइलिन, ये इथाइलिन के अगेंस्ट काम करता है. सेकंड इज साइटोकाइनिन, साइटोकाइनिन भी सेम ऐसे ही काम करता है. As we have discussed that we can produce off-season flowers, so in same way we can produce off-seasons vegetables and fruits that we often see in the market. जो ऑफ-सीजन क्रॉप्स हैं और वेजिटेबल्स हैं, फ्रूट्स हैं, वो हमें मिलते हैं प्लांट फिजियोलॉजी की नॉलेज से. सो सेम वे इसमें भी दो कंसेप्ट काम करते हैं, दैट इज़ फोटोपीरियडिज्म एंड वर्नलाइजेशन. So these two factors plays a very important role in getting off-season vegetables and fruits. So important source of farmers' income, और off-season में जो returns हैं farmer के लिए बहुत अच्छे मिलते हैं. So this is produced by using knowledge of photoperiodism and vernalization that I have already discussed. So yield of leafy vegetables can be enhanced by providing them more light. So root crops like we have radish that we already grow in our kitchen gardens, so give yield better yield under short day condition. वैसे जो रेडिश है, दिस इज़ अ लॉन्ग डे प्लांट, इसमें फ्लावरिंग लॉन्ग डे कंडीशंस में होती है, बट वी ग्रो इट मोस्टली इन शॉर्ट डे कंडीशन. सर्दियों में से हम ग्रो करते हैं, एंड इन विंटर्स इट इज़ अनेबल टू प्रोड्यूस फ्लावर्स. फ्लावर्स में प्रोड्यूस नहीं होते हैं, एंड वी रिक्वायर ओनली वेजिटेटिव पार्ट. सो वेजिटेटिव पार्ट को ही हम कंज्यूम करते हैं. सो दैट्स व्हाई दिस इज़ वेरी इंपॉर्टेंट टू ग्रो दिस क्रॉप इन शॉर्ट डे कंडीशंस और विंटर्स. Cucumber, that is Cucumis sativa. So it produces more staminate flower. Staminate flower means male flowers. जो मेल फ्लावर्स हैं वो ज़्यादा प्रोड्यूस होते हैं ड्यूरिंग लॉन्ग डेज, और जो मेल फ्लावर्स हैं दे आर अनेबल टू प्रोड्यूस फ्रूट में कन्वर्ट नहीं होते हैं वो. एंड बाय रिड्यूसिंग लाइट आवर्स, अगर हम लाइट आवर्स को रिड्यूस करते हैं, सो वी कैन इंड्यूस टू प्रोड्यूस मोर फीमेल फ्लावर्स, और जो बेसिकली फ्रूट्स में कन्वर्ट होते हैं. So by reducing the light hours, we can produce or induce the plant to produce more female flowers so that we can get more fruits. So these PGRs can be used for improving the growth of the plant. Like we have this GA, so it is used to enhance the shoot growth. ये shoot growth को improve करता है. Then PGRs like putrescine, brasinolide, salicylic acid, resorcinol, ये भी as a stimulant use होते हैं, and they can improve the growth of the plant. Then we have digulic and pelcobutrazole. It increases side branching, जिससे कि branching increase होती है, and we can get more yield or higher yield. Then GA used to spray tree seedling in the greenhouses to enhance vegetative growth. जो GA है, that is gibberellic acid, ये vegetative growth को improve करता है, means plant की growth को improve करता है vegetatively. The next, apical dominance suppression. First of all, what is apical dominance? जब किसी प्लांट में एपिकल बर्ड प्रेजेंट होता है तो उसकी जो लिटरल ब्रांचेस हैं वो ग्रो नहीं कर पाती है. सो दिस इफेक्ट इज़ नॉन एज एपिकल डोमिनेंस. सो ड्यू टू प्रेजेंस ऑफ दिस एपिकल बर्ड there is suppression of lateral growth of these axillary birds. So this suppression of lateral birds or axillary birds due to presence of this apical bud is known as apical dominance, and this is due to one important hormone that is auxin. Auxin की वजह से apical dominance होती है और plant सिर्फ height में grow करता है. If we remove this tip, then there is formation of axillary bud और ज़्यादा branching बनती है इसमें. So this is the suppression of apical dominance. This chemical that is digulic, it is used to suppress the apical dominance. ये apical dominance को suppress करता है और plants में बहुत सारी branches produce होंगी, axillary buds भी grow करेंगे. So here we can see here. So this is apical bud. Basically we can see here, if we remove this apical portion, so there is these axillary buds can grow. और ये जो suppression है इसकी वजह से इसकी, that is called apical dominance. If we remove this apical bud or this apical portion, so there is growth in this axillary bud. The next is dormancy breaking. This is very important. Dormancy एक condition होती है when seed or bud fails to germinate or produce leaves or flowers under suitable conditions. So that is called dormancy. अगर seed को हम proper conditions दे रहे हैं पर फिर भी seed germinate नहीं कर रहा, that is called seed dormancy. So it can be this problem can be solved by using certain PGRs जो कि seed dormancy को break करते हैं. The one of the best is this GA and cytokinins. So hindrance in germination of seed is known as dormancy. जो seeds germinate नहीं कर पाती, that is called seed dormancy. So we can use certain cytokinins. जो cytokinin है, this is natural hormone. इसके कुछ synthetic chemicals भी हैं, like we have benzyl adenine or we can say benzyl aminopurine, abbreviated as BAP. This is a kinetin or cytokinin, and this is used to break dormancy. Then this chemical also helps in delaying senescence. जो aging process है plant में उसको भी ये delay करता है, जिससे जो leaves हैं वो green रहती हैं और ये yellowish नहीं होती है और abscission नहीं होता है. So that aging process can be controlled by using these PGRs. Then we have naphthyl acetic acid, NAA, and putrescine. These are reported to delay and suppress the senescence. ये भी senescence को prevent करते हैं. Next is suppression of transpiration. So transpiration, loss of water होता है from the apical or aerial part of the plants. जो aerial part है plants का, in the form of water vapor, that is called transpiration. So we can use certain chemicals which are known as anti-transpirants. जो transpiration को रोकते हैं उन्हें कहते हैं anti-transpirants. So ABA, this is one of the most common plant hormone. This is also known as water stress hormone. इसे water stress hormone भी कहते हैं, जो कि स्टोमेटा को क्लोज करता है और ट्रांसपिरेशन को स्टॉप करता है. देन वी हैव दिस ट्रिपल सी, दैट इज क्लोरीन क्वेट क्लोराइड. दीज केमिकल्स आर रिपोर्टेड एज एंटी ट्रांसपेरेंट्स. ये ट्रांसपिरेशन को रोकते हैं. दिस केमिकल दैट इज क्लोरीन क्वेट क्लोराइड, ये भी स्प्रेस करता है ट्रांसपिरेशन को. दिस इज यूज फॉर इंक्रीजिंग यील्ड ऑफ क्रॉप प्लांट्स. द नेक्स्ट हर्बिसाइड्स. हर्बिसाइड्स ऐसे केमिकल्स होते हैं विच आर यूज टू किल द हर्ब्स और वी कैन से वीड्स. जो अनवांटेड वीड्स हैं उनको किल करने के लिए हम केमिकल्स यूज़ करते हैं, उन्हें कहते हैं हर्बिसाइड्स. सो दिस इज द मोस्ट कॉमनली यूज हर्बिसाइड. ग्लाइफोसेट, ये अब बैन हो गया है. दिस केमिकल हैज़ बीन बैन्ड बाय द गवर्नमेंट. सो PGR, like we have this 2,4-D. There was one agent that is known as chemical agent that is known as Agent Orange. This chemical was used in Vietnam War by US military. Vietnam War by US military. So this is a combination of this 2,4-D and 2,4,5-T. तो दो केमिकल्स हैं, उनके कम्बिनेशन को कहा जाता है और एजेंट ऑरेंज. तो ये यूज़ हुआ था वियतनाम वॉर में एंड इट एक्ट एज अ डीफोलिएटिंग एजेंट. ये पत्तों को गिरा देता है प्लांट से. दिस इज अ डीफोलिएटिंग एजेंट. सो इन एडिशन टू दिस वी हैव दिस डाइक्वेट एंड पैराक्वेट. ये जो केमिकल्स हैं, दीज आर यूज्ड एज हर्बिसाइड्स. मीन्स ये जो वीड्स हैं, हर्ब्स हैं, उनको किल करते हैं. दिस पैराक्वेट. Then next is defoliation, means removal of leaves from the plant. That is called defoliation. So ABA can be used as a spray for removal of leaves from the plant. So ये जो leaves गिर जाते हैं, that is called defoliation. The next, growth retardant. So certain chemicals or PGRs can stop the growth of the plant or reduce the growth of the plant. So we have some chemicals. So ABA, this is ethylene, diquat. Then we have fluorinol, glyphosine, malichyhydrazide, palcobutrazole, tibba. These all are potential growth retardants. तो ये growth को stop कर सकते हैं. These are used to induce flowering by cutting short the vegetative phase. ये vegetative phase को कम करते हैं, means जो plant है वो early stage में flowers को induce करेगा. So they can cut short the vegetative phase in the plant. So this was all about for our discussion about plant physiology, its aims and objectives, its application in agriculture and horticulture. Hope you'll get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in comment section. Thanks for watching. Have a great time ahead.